Hi everyone, my name is Samira and my work is entitled Acute Exposure to Gallic Acid Induces Anxiolytic Behavior in Zebrafish. So, gallic acid is a plant-derived polyphenol that, when metabolized, your metabolites known as gallates. They easily cross the blood-brain barrier. But you let me know a little about the structure of gallic acid. So the structure of gallic acid consists of an aromatic ring with the three polyphenol groups here, the OH and a COOH group. And in this polyphenol group here, the OH, the planar arrangements confer the property of a free radical scavenging to gallic acid. But why analyze gallic acid? The gallic acid is an intermediate key in the synthesis of hydrolyzable tannins from plants. It is also an anti-inflammatory and the primary cardioprotective agent found in wine, green tea, and cocoa. A little parenthesis here, the most agent found in green tea wasn't gallic acid, was your metabolite known as epigallocatechin, but we wasn't studied in this presentation. So the potential of the gallic acid, the antioxidant potential, depends on some factors, such as the concentration of gallic acid, the iron content, and the pH of the body. And the gallic acid is used in processed food and cosmetic and food packing materials to prevent rancidity induced by lipid peroxidation. But gallic acid can be found in your alimentation. These examples are from plants found in Brazil, and the food can be cajá, the plant found in the north of the country, and cajueiro, it's found in the north of the country too. But, however, it is so many studies using gallic acid, as you see here. There is studies analyze the approach anti-inflammatory of gallic acid, analgesic, antiviral, anticarcinogenic, and anti-apoptotic. But I will focus in antioxidant potential of gallic acid. But all these studies doesn't use zebrafish to analyze the gallic acid. They use murine models or invertebrate models or in vitro models too. But maybe it could be important to analyze use zebrafish behavior using gallic acid to analyze your behavior of the gallic acid and the impact of gallic acid in the brain. Maybe in the future could be used for neurodegenerative diseases like Alzheimer or Parkinson or stress or anxiety problems because the potential of gallic acid. Now I'll explain a little bit how works the novel tank test. So the novel tank test consists in a trapezoid aquarium with divided in three different zones virtually, which is top, middle, and bottom zone. And dependent on the time the animal is spent in these zones, he has a different behavior. When zebrafish spends most time in top, he presents a behavior, anxiolytic behavior. When he is spent in the bottom, he have a anxiogenic behavior. So the objective of the studies was evaluate if GA is able to alter the locomotor and exploratory profiles and induce anxiety or anxiogenic-like behavior profiles in zebrafish through the novel tank test. So the method was, we decided to make this work in, divide the animals in 4 groups, which is control, gallic acid with 5 milligrams per liter, gallic acid with 10 milligrams per liter, and gallic acid with 20 milligrams per liter. And we use 12 animals per group with random sex mixed. And why you do this? We decide put the animals in a cryo treatment for 24 hours, exposed to the different concentrations of gallic acid. And after the end of the exposure, we start the novel tank test. We recorded the animals for 6 minutes each, using ANYmaze software to make the track plot to see how the zebrafish the behavior works. So now, after our analysis, we have some results. So, as you can see here, this is a result for the locomotor profile of zebrafish. As you can see, didn't have any significant change in distance or mean speed or line cross of the zebrafish. So gallic acid wasn't capable to alter the locomotor profile of the zebrafish. But in relation to exploratory profile, zebrafish have some change, which is the time the animal spent in the top zone was the highest concentration of this gallic acid. When you compare the concentration against the control, there is a significant difference. And when you compare the 20 milligrams per liter of gallic acid, when you compare to another lower concentrations. As the same for the number of entries of the gallic acid when you compare to the control and when you compare to another concentrations of gallic acid. But in relation to the time active, we have the same results. The gallic acid, the highest concentration, was capable to let the animal exploring the zone. As you can see, they didn't have the alteration in locomotor profile, so the animal didn't spend freezing in the zone. In relation to the middle zone, have almost the same results found in the top zone. There is a difference in the time here when you see the highest concentration of gallic acid when you compare against the control and against it to another exposure group. And in relation to the number of entries, the gallic acid was capable to alter the number of entries, almost 23 entries when you compare to the control. And he spent your time active in the middle zone too. And in relation to the bottom, the zebrafish spent the highest concentration. He spent less time when you compare to control and against to two another concentrations. Now I make a nutshell. As you can see, this is a model of novel tank test, and the highest concentration of gallic acid, which is gallic acid with 20 milligrams per liter, he spent almost time here, and he didn't spend more time in the middle and bottom. The other two lowest concentration, the zebrafish explored only to the zones. And the zebrafish make more change of the number of entries, only the zone, the top in the middle, and he always make this, as it was, you see the graphic a few minutes ago. And in so conclusion, the concentration of the 20 milligrams per liter of gallic acid, he was capable to generate an anxiolytic profile in zebrafish. Can be possible when we increase the concentration of gallic acid, zebrafish will spend almost every time of the test in the top zone. And for the next steps for this work is analyze other parameters such as preference test and biochemistry analysis such as stress oxidative or analyze another neurotransmitter system, signaling. And I would like to acknowledgement my lab, the people who helped me to make this work can be possible. And I would like to acknowledgement the CAPS that make my fellowship. And that's it. Thank you for watching.